whatever we are doing, attempt and using a technology to enhance the efficacy and reduce the side effects. And we have already made it very much clear, any therapeutic agent we use it now, it has got some sort of side effects. It is bound to be present. So we are going to create a illness to treat a illness. So in order to avoid that, to give a additional, it is safer and it is effectively getting used in the given timeline. So we need to use this, adapt the system. There is no other option. Even though the technologies are very tougher and costly, we need to adapt because they have got inherent advantages. But we should not misuse it. That is only the danger which is humankind is having now. So people are inventing a problem and keeping a solution with them and launching a solution and earning the money. Let it be. So we are going to have that concept so that we will be making further cheaper those medicines so that it will be available to the society. Now, we are entering into a little bit concept. We are discussing about a little bit terminology of what we are going to have in the previous class. We got end there. So now we are entering into the nucleic acid therapeutic delivery systems. Most of the times they are all going to be used. Now we are entering into the different than target delivery. Now gene therapy. So the gene therapy is the approach, which is actually a targeting only. We are targeting to the nucleus, and at the molecular level we are targeting it. So even it is not targeting at a nucleus, we are targeting at chromosome. It is not only the chromosome, it is the specific segment in the chromosome. So this much specific targeting we are going to do. They are very much required in the gene therapy. So now we are entering into a little bit concepts of a gene therapy, and we are going to a little bit go advance in nearby future. I hope you are able to see the screen and you are able to listen to me. So if you are having any voice break, just unmute yourself and say, so that I will be able to repeat again certain slides which I am having that particular problem from my end. So in this particular case, we have already seen so many delivery systems. We have already discussed to launch these moieties specifically to the site, like nanoparticles. We have seen aptamers. We have seen fullerenes. We have got carbon tubes. We have got quantum dots and gold nanoparticles. So these are all the things which is making delivery systems by means of using polymers, or it may be using minerals. So we are able to do them. And now in our syllabus specifically, it is mentioned that Liposomes has a drug delivery system, because instead of having all the things in the syllabus, we have got one point specifically which is present in the Liposomes as a delivery systems. So in the gene therapy, we have got a specificity and how the liposomes are used, and why it has got a ability to use them as a specifically when all different types of nano carriers are available, why they have been chosen and given in the syllabus. So this is actually our concern, and we will introduce this particular topic now, and you will be getting a 5 marks question or 10 marks question. What are the effect of liposomes, and what is the use of liposomes in the drug delivery, or it may be a drug targeting, or it may be a gene therapy. So these are all the questions you are going to be expected in the exam. Later we will go further depth in different types of gene therapy, and one we are going to cover now, and remaining things which we are going to deal later. So that is actually a gene therapy systems a little bit complicated and advanced. We will see later, because liposomes we are frequently listening and we know the concept, so we are entering it now. So the liposomes in the gene therapy, most of the times we know that the gene therapy is a therapeutic approach. This is what we are going to use is treating a disease. Therapy means a person or a patient, he has got some problem. We want to give a treatment to him, so which is having a beneficial effect when compared to harmful effects. I have already told you we are having both inbuilt. When the therapeutic benefits are getting more and more overweight, and we take the compromise to launch that particular moiety into the market, because immediately we have got a bigger problem. So if that bigger problem is solved, even smaller problem is also there, we will manage later, or our body will manage in due course of time. This is actually what we are having a concept called as a drawing a bigger line on a smaller line, so that the smaller line will appear, even though it is previously it is bigger, it will appear as a smaller. It is somewhere, it is also happening here. Sometimes in the misfelt or it is a wrong technology, it may cause the damage to that extent. The smaller line will become further bigger. So give the further problem also. So that is the reason we need to be very conscious, because so in this we are going to deliver the genetic material into the cell. So the basic factory of a cell plant, we are saying that in each cell in our body, 3 million trillion cells are present in our body. Each cell is a bigger factory. Like we have got a pharmaceutical industry in a particular location of any city. So the same thing which is going on within the cell also. Then complicated moieties which you cannot prepare outside. The cell itself it is preparing within a teeny moiety, within a teeny space, with a teeny resources, with a whatever you say, the size is very teeny and very small. It is not visible to the naked eye. So your human brain, and you have got a trillions of the cells, you have got plenty of the space outside. Even you face a lot of problems to synthesize the similar moieties. What the cell is synthesizing and doing, it is not possible. That is the reason. Even though we think that we are the highly intelligent animals and we are high intelligent in this universe, but even cell function, what we are doing and seeing, and we are able to study, we cannot mimic that. It means our intelligence is not that much extent that the universe or the creator of this universe has made them inserted into the cell. So what we know after knowing all these things is, what we know is only teeny, a minuscule of whatever which is present as existing knowledge. So it is very tough to understand what the journey is going on, how that cell is made to do all these activities, who is guiding it, and how it is getting triggered, who is making it to die. So what is happening? So these are all the questions. Some are answered and some are unanswered. These unanswered questions are resulting into some diseases. So these diseases are again becoming as a questions. So mankind is finding an answer to treat them. So in this journey, this progress has happened, and we have reached to the molecular pharmaceutics to level of treating the gene, where the problem is actually originated. In fact, what we are taking now is maybe at the gene level we are having. We need to go further, deeper and deeper there. We may be having at a quantum level, and we are going further and deeper and deeper. Maybe we are going to have in the form of a different dimension, so which cannot be even possible to use this materialistic equipment or plant, may not be able to synthesize it. There is a journey which is of non-materialistic approach. It may come. These are all the different steps, and our Indian philosophy has made it all much clear in the doctrines which are present in the Indian philosophy. They have given a lot of answer to these questions. And if you are going to little bit deep in Veda and Upanishads, around 10 or 11 are present. If you ask the answers in it, you will get plenty of answers. But those answers are not will not be possible in the materialistic world by using what our physical and chemical principles. They are of a different dimension which has already been given. Let us see, our mankind will come to that particular level where the science are able to do by seeing and touching and perceiving and thinking, and be able to treat the diseases. It is not using any means of physical matter to treat the disease. So this is actually of a different dimension and going to the level of a futuristic approach for the gene therapy. We expect at a similar level. The world is seeking that, yes, how Indian philosophy and made it a long back, whether it is a grafting or it is a treatment of a disease, and without having a physical in front of them and making from a longer distance, they are getting treated. How it could be possible? So these are all the questions we need to answer and going to the particular journey. Now, in this particular case, we are dealing now in our curriculum is the gene therapy. In the gene therapy, we are attacking a genetic material. So we know that the genetic material is DNA and RNA. Preferably, this is two things which are having and making a differences and uniqueness in this particular universe creation. So we are going to attack them. It means it has got intrinsic capabilities. They have been made by themselves by having a protectant layer not to get disturbed by the external agency, and they have got antisense genes, so that they will be generating a concept of a templates, and the templates only be getting disturbed but not the original genome. So this is actually another protection mechanism which is being made by the creator, so which is making it tougher for our journey to enter there and reach there and correct that. So this is another problem, because we are dealing genetic material like DNA and RNA. when it is used in the therapeutic reagent in the gene therapy, they are required to deliver a sufficient quantities of the correct target sites, sufficient quantities. Why we want to? We need to give a treatment which is in a sufficient quantities. At the same time, it is in the target site to achieve the desired level of therapeutic effect. It should not be lower, it should not be under. So the desired level of therapeutic for that desired time frame. Why this time frame is very much necessary? Because we want to make our generation of this particular human being getting treated, or next generation should also be having it treated. This is actually what the time frame of our gene therapy is. So we are going to see a little bit later how this is going to affect our gene therapy to the offspring. Means next generation of the patient, we are treating them, because we can make the time frame which is restricted only to that particular patient. In that particular case, we don't want to touch anything which is having a genetic material as a basic function, and we are not going to deal and touch with it. And we want to make the treatment in that particular human being as a patient, as a separate entity, and the offspring will not be affected. But in the different time frame, we don't want to make next generation to suffer the same disease. In that particular case, we make a treatment in such a way that the entire correction, it will produce the offspring without the particular problem. This is actually what the world called as the desired time frame. And also the design time frame is, we need to make it very fast. And because of that reason only, we have got so many polymerase reaction, PCR reaction, RT-PCR, we are saying everything. So we have got how many number of cycles we are doing. Maybe it will be 26 cycles or 30 to 80. So how many number of cycles which are making it? That's what we are telling, that whether corona is present or not. When number of cycles are lesser and lesser, it means the viral load is more and more. And RT-PCR, how many cycles they have done, that will give the idea that how much extent the person got affected in the given time frame, like 48 hours, 2 days, like that only. If it is taking years to replicate them, then it is not good. Means the patient will die before we are diagnosing it and making a medicine and treating it. It means it is a complex, but we need to make it a simple. Even though it is unachievable, you are making it possible, and we are making it a time span in the short duration, so that we are going to have a treatment to the patient ready. So this is actually what the desired time frame means in the angle of generations, in the angle of within the generation, what the treatment period is. So in this particular case, liposome is one of the promising vector. We have got a biological vector, in vivo vectors, and means non-bio vectors. So we have got viral vectors and non-viral vectors. So the liposome is one of the non-viral vector. It is not belonging to any species of virus like that to deliver the gene. So it is one of the promising vector to deliver the molecular cargo. I am calling it as a cargo, means we are sending our cargo to different countries in the aeroplane. So you know how huge the aeroplane is. It will be taking around 10 buses. If you see in the recent, when you have got oxygen from the different countries in the cargo, the trucks has got entered into the flight. So this is actually entire trucks got unloaded from the flights. So it is reached to the different places by the train. In the train, they have got super flat top and made the trucks go there. Ultimately, we got reached in our Bharatanagar station recently, the oxygen. So how these bigger macro stages of your truck-sized material which has got, now we are making at the molecular level, because that is the reason we are calling it as a small gene therapy, which is present as a molecular cargo. That the molecular cargo in our case is very teeny, that is like a DNA and RNA for the therapeutic benefit. So this is actually what we are going to nanonization. We are going to coming from the macro level outside to we are going within and within. Why we are doing it? When you are going attacking within and within, so the treatment is becoming very faster and more specific and having a lesser side effect, and not much time is required. Because if you are taking a macro molecules or bigger cargo, it may take the days, and reaching here it may be weeks, but it may take in this particular molecular cargo, if you are doing it perfectly, it may reach within minutes to the desired site. So this is actually what the called as a nanotechnology. We are representing the word nano. We are going to nanonization in our delivery systems also. So it is mean that nearby future we are going to have a delivery system. It is going to be made most of the times with invisible only. Invisible means everything is making it very teeny. With them only we are going to handle. So unlike these viral vectors, the liposome-based vector can be designed actively, that is active for the active passing also, and specifically target site via targeting molecules. Whatever we want to reach, most of the times it is uh, nucleus we are going to target. Sometimes it may be a cytoplasm of the cell. That is our endpoint in our journey. So depending upon the what it is disease we want to treat it. We have already mentioned, if you want to make our offsprings or the next generations not to be getting disturbed, our journey may be ending at cytoplasmic level. So we want to treat the next generations also. We need to treat at the cellular level of a nucleus. So at the basic stem cell place, that is a space where we want to treat. Then we may be required to enter nucleus also. And specifically, we need to treat the chromosome portion also. So this is actually what our journey is. So if you see here how this non-viral gene delivery by the non-biomolecules, non-viral molecules like a polyplex and lipoplex. These are all the complex polysaccharides or complex polymers. They group together. So we are generally naming them as neosomes and liposomes. So these liposomes and neosomes, which are making a journey, so that we are able to take the material of a desired moiety to the desired site. Because of that one, we are able to have these complexes are made up of the lipoproteins and the lipopolysaccharides, and we are going to have a polymers to reach the desired site without using any virus. Because virus itself has got some virulence. So if these non-viral vectors, they don't have any virulence. Only the thing is they are foreign to our body, so they may be getting removed. That is only the problem. So that is what we need to avoid. So for that purpose, what we are doing, say for example, we are taking a cationic liposome or a cationic polymer, a neosome, so to entrap our DNA molecule. So this is actually what we are making a teeny delivery system, which is visible only to the electron microscope. It is not possible even making it seen by our ordinary compound microscope. So this is actually what we are making a condensation process and making a polyplex or lipoplex. And because this is a lipopolysaccharide, and if it is considered by the cell as a foreign to our body and it is not equal to our body material, it is having a change. And it is having a lipophilic in nature, and polymers are also having a longer chains. They are also having a lipophilic in nature. It causes the endocytosis. Endocytosis means they engulf. In order to kill that, the cell will engulf it. So this is also named as a phagocytosis. Then what will happen? These polyplexes and lipoplexes, they enter to the cytoplasm. So they will be converting it as the endosomes. So they will be getting attacked with lyosomes. They are the polymer, they are the enzymes, they cleave the moieties and liberate the DNA fragments because they fragment it, because their body is considered them as a foreign. So they cleave it, and the DNA fragment will cause, and it causes the DNA degradation, and the fragment will be floating in the cytoplasm. They may be using for a precursors for the next journey. So it is actually got wasted. Whatever our time and efforts, what we have done is wasted. This is actually what lysosome attack after the phagocytosis. So sometimes what will happen, this lysosome attack will be gone, then will be having endosome escape. So they will be having the DNA nanoparticle, and after that they are liberating into a individual DNA. Sometimes what will happen, they will be fragmented outside itself as a DNA, whatever we have loaded there, and that DNA again they will be having a problem of these enzymes. They will again degrade. It means if we are taking a targeting, if you are able to reach these particular polyplex and lipoplexes, certainly entering into the nucleus, your journey of reaching to the nucleus is achieved. That is, your targeting index will be very higher when compared to a normal conventional delivery system. This is what we are doing here. So we are making our journey of entering of these liposomes to entering into the nucleus. So how many liposomes we are making it and which are loaded with DNA reach into our nucleus. So the total nucleus when it is reached, how much it is reached, and when compared to what you are giving, so the number is more. It means we are efficient. When it is compared to your conventional dosage form, if your targeting is resulting more and more, then your targeting index is better and better. Then what will happen? What you are going to take advantage? This DNA is liberated within the nucleus, and it will serve some purpose there. It may be a correction, or it may be making a change, or it may be causing a mutation, or it may be getting inserted, and it will cause the transcription, and the transcription will be the RNA. That RNA will translate into a desired protein. Because previously, when the disease is having, that protein is not getting generated, that the patient is suffering. Now it is got corrected. If you see the extreme right side, the mRNA after the treatment is correct, mRNA is getting released. That messenger RNA, it will act as a template for the translation, where the tRNA, that is transfer RNA, will be using and making a the codon converting the anticodon, and this anticodon is making it into a protein moiety sequenced by using amino acids. So you know that all the proteins are made up of amino acids, whereas this RNA, DNA is made up of nucleic acids. So these sequences which are going to be liberated in the cytoplasm, the proteins are rich in that protein in a sufficient concentration, then they will start doing their job. Previously, that is getting depleted. The protein may be insulin, and the person is taking an insulin regularly. Now, he may not require it because the factory is already installed within the nucleus. Previously, the factory got shut down. Sorry, we made it inserted in the particular cell. Now the cell itself acting as a factory to synthesize this protein, and it is actually giving to the patient. So he need not getting pain by taking repeated insulin injections in the morning and evening throughout a week and 365 days. So it is highly painful technique to regularly take. Even if you skip it, it may cause the damage of changes in the glucose level. It is one diabetic most prevalent one. Like that, we have got so many treatments we can make it by releasing it a proper protein, by having this insertion of a DNA taking to the targeting to your nucleus and making a journey successful. In this particular journey, what will happen? Even if you have got a failure, then what will happen? Your DNA fragment is nothing but a nucleic acid moieties. They are precursors for our growth, so they will be used. Only the disease will not be cured. It is not but much damage. Your polymer, they are highly GRAS listed, and they are generally accepted as regarded as a safe materials. So that will not cause much damage. And similarly, the polymers and the liposomes materials, like your phosphatidylcholine, or it may be you are using a cholesterol, and all of them are very safer one. They will not cause a damage. So your side effects will not be much. Even if it is failure, you will not be having a treatment of a disease, but you will not be getting affected with much side effects. And if it is treated, it is efficacy will be there without having a side effect. This is actually what the beauty of this journey. This is the reason these liposomes have been considered as a very good moieties to treat. We will see why it is having, because in your 5 marks and 10 marks question in the exam, you need to write a little bit deeper. We will see what is the advantages of liposomes for the gene therapy. The liposomes are considered an ideal drug carrier system since why? Their morphology is similar to the cellular membrane. This is actually what the beauty of it. The delivery system when compared to remaining things like nanoparticles, you got maybe a dendrimer, it may be a quantum dot, or it may be a gold nanoparticle, it is a fullerene, or it may be any carbon nanotube. In all the cases, it is not like that. A cellular membrane-like structure is not there, except in the liposome. Even in the neosome also, you don't have it. It is completely a chemical polymer. We are making it similar to our biological membrane. It is not same. It is appearing as a similar. So this is the reason these liposomes are considered when compared to remaining nanoparticulate drug delivery systems for the targeting, and it is being used successfully. That is the reason we are finding them in the liposomal systems in market also. So due to their properties, and liposomes deliver not only nucleic acids, of course we have got a specific concern there for to target nucleus, but also other target ligands like peptides and proteins, that is antibodies, your aptamers. So they are actually nucleic acids that nothing to do with transcription and translation. Just they are tagged to have a specificity taken to the cellular surface. That is actually what we have already learned in the terminology as aptamer. It is also same as that of the small fragments of your appearing as the nucleic acid sequences, and they will be, because of that one, they will be having a specificity towards particular receptor which is present in the cell. So we are going to use them, and we can tag them onto these liposomes in addition to our folic acid. Most of the times you may be remembering that a pregnant woman will be taken more levels of the folic acid in the first trimester. It will be given because where the nerve cells are getting formed very high frequency, and we need to supply the raw material. The folic acid is given in the pregnant woman. So we need to make this availability there. So we are making a journey, and we are able to use them for the wide variety of the molecules, whether it is a lipophilic or lipophobic, that is hydrophilic or lipophilic moieties. So the efficacy of the liposomes as a carrier systems depend on the physicochemical properties of their membranes. So what we are tagging on them, so the nature of their components and their sizes and their surface charge and the lipid organization, whether it is a monolamellar, it is multilamellar, it is flexible, or maybe it is a strong or unsheathed, or what we have done. Because of that, that efficiency will be depending, and the targeting index will be varying. What are the liposomal types? We can make them so that we will be reaching different levels of drug targeting. This is actually what has been made by a human being, an attempt to have a wide varieties of the chemicals which are tagged to them, and we are attached to them or inserted in them, so that we will be having a different types of nano liposomes, and we are going to have a different advantage. So the chemical modification of the liposome can efficiently improve their performance in terms of circulation longevity. This is actually what is very much important. Even it is leaching out and diffused out of your cell membrane and circulating within the blood, but it has got a longer duration of action. Again, they may have the chance to uptaken by the similar cells, and they increase the efficiency. This is one. And target specific delivery. We have made the surface of this liposome chemically modified. We can have, like we have attached with the aptamer, then it has got a specificity and enhanced intracellular penetration, because when your drug delivery system is not reaching the cytoplasm, how can it enter nucleus? So it is not possible. So we are going to enhance the intracellular penetration by having the modifications onto the surfaces, and contrast enhancement for the image guided therapy in the diagnosis. And we want to see that our targeting is proper or not. We will be tagging with any radio ligand, radioactive moieties, or maybe having a fluorescent guided chemicals, so that we can see them whether that the target is getting loaded with this or not in the diagnosis, and to find the evaluation and analyzing the efficiency of the drug delivery system. In that case also, we can be able to do it. It means these liposomal times not only having in the therapy, it is also having in the diagnosis. Not only that, it is in the drug development research, where we have got developing a drug where it is reaching to the target site or not by having a image guided principle, so that we will be able to see initially in the experimental animals whether it is having a specificity or not by having this image guided therapy. Later onwards, we will remove those radioactive or the fluorescent material from the liposomal surface, and we give the similar one and see that whether it is acting or not. For that purpose, we have got a clinical trials, and if the clinical trials also showing that in the similar specificity, then we are going to have, yes, this is having a successful in nature for the gene therapy. If it is not successful, we need to create a stimuli sensitivity. The stimuli sensitivity may be either it is an enzyme sensitivity, or it may be a pH, or it may be a specific receptor. So these are all things we need to find out and tag the liposome with them, with those enzymes. So they will be modifying it accordingly need, and they sense it and going there and attacking there and reaching the site without any problem. So this is actually what the chemical modification is making a wide varieties so that we will be having the advantages. The success of the liposomal mediated gene delivery is a multifactorial issue because it is a complex one. If you are employing a DOE, in the case of liposomal treatment, it has got a wide variety of issues. In the case of your excipient selection, in the modern pharmaceutics, we have seen drug excipient analysis. So in the excipient selection, and we have got a process parameters, and when we have got a size related and effect of material and the formulation related parameters, and also we have got biologically related parameters. At the same time, we have got your reticular endothelium, that is immune system related and specificity related parameters. If you see, your variables of independent variables are keep on increasing. Only the dependent variable is concentration of the particular one in the desired site. To achieve that, we have got a wide variety of independent variables. So we have got a Alvi-Prasad principle. So where we have got a multifactorial designs, if L to the power of V, number of levels, if you even you are taking 2. So when levels are increasing and the factors are getting increased, in this particular gene therapy, we have got piling up so many factors within the cell and outside the cell and outside the factory and within the factory, and process related and formulation related and material related. So these are all getting together, it will having a n more, that will become a multifactorial issue. That is the reason well-designed liposomal system might lead to well-designed. This is what we are making a project. We are going to design well so that I am making a formulation which are having very less number, but reaching my target. With less number of formulations, I am going to make it. This is actually what the well-designed liposomal systems might lead to the optimized gene transfection. It means it is entering into the either it is a cell or it is a nucleus, especially in vivo. Sometimes what the biggest issue is, it may work in ex vivo. Ex vivo means outside the body, they will work. And we will be very happy with the result. That's what we do in our lab, in the projects. We do only in the ex vivo. But when we do the same thing in vivo, it will not work. So we need to make this optimized gene performs in the in vivo system also. This is actually what the end of journey. For that purpose only, we need to make a different liposomal types and see that which is reaching better, and we need to finalize that and optimize it. To date, the liposomes with different characters have been applied as a drug carrier, such as the PEG bearing, that is actually polyethylene glycol, and we are always the buzzword is the pegylation or pegylation, and charged liposomes, specifically whether it is anionic or cationic, and stimuli responses liposomes. So these are all the different types we are making it. One is PEG bearing liposomes to bypass your reticuloendothelial system, and charged liposomes, we have got a specificity on the surface. Even that specificity is not getting managed, we have got a stimuli responsive liposomes. These three approaches which are having, even in the stimuli responses, we have got a subtypes like a stimuli of a pH, stimulum of a particular enzyme. So stimuli of a chemical or it is a charge variation or oxidative and reductive principles. So these are all acting as a stimuli to give a response to trigger the release and the transfer. That is all. That is actually a wide varieties are present now. So we need to make them different types and see that which type is working better, and we will be finalizing, and at the end we are optimizing and launching into the market. If you see here, what are these designs which you can make it in a nutshell in a single slide. If you see here, what is happening, this lipid bilayer which is present here in the screen, we have got yellow dots where we have got this hydrophobic, that is hydrophilic heads are facing. and the tiles are present in the center, that is blue zone. It is in the center, that is in the lipophilic in nature. So this is actually lipid bilayer, which is semi-present, same as that of our cellular, mimicking the cellular structure. So it has got outside, there are two types of the resins are present. One is called as a high PEG density, another one is called as a low PEG density. Generally, the high PEG density, which is having a longer length and which is having around 5 nanometers. Most of the times, which we have already seen that these liposomes are having in the size of 2 to 100 nanometers, which you are having different depending upon. Most of the times it is 20 nanometers like that diameter. So you have got a brush resin which is having high polymer density with 5 nanometers thickness, whereas the mushroom design, which is having a low polyglycan density, which is having a 3.5 nanometers thickness around them. So this is actually creating a lipophilic moiety enhanced around the surface of these liposomes. So here, after making it, we have got a different sites which you can load the drug. One is within the cellular membrane, that is liposomal membrane, and inside the membrane on the surface of the membrane. One is on the side surface, or within the membrane, or inside the membrane. It means endo, exo, and within that. So you are going to have these loading which will be taking place. So this, if you see in the center, you have got 3 red dots. They are the hydrophilic drugs. They are getting entrapped in the water which is entrapped within the liposome. So this is what it is a good site to encourage your drugs which are in the hydrophilic in nature, and they will be sheathed with lipophilic membrane. That is the reason they behave like the outside it is behaving as a lipophilic in nature, but inside drug is hydrophilic in nature. It is carrying and it is traveling along with the liposome. This is actually what the beauty of this type of concept we are going to have. This approach we are going to use in the design of drugs which are having a water solubility very high, like your class 1 drugs. Most of the times you are having this and you have got a class 3 drugs. Water solubility very high. But you have got a class 2 and class 4 also, where the liposolubility will be there in the BCS classification. There you have got in the cell membrane, in the blue region of your liposome, you have got 2 red balls are present. That is actually called region for the loading of hydrophobic drugs. So these hydrophobic drugs are getting dissolved in the membrane, and we are going to taking them to different sites. So this is very good for the loading of your class 2 and class 4 drugs, where they are having a lipid solubility and low water solubility. So hydrophobic drugs, that is the lipid soluble drugs which are entrapping in the membrane itself. And there is a third approach they will be having is conjugating onto the surface, where it is a lipophilic and a zwitterionic like, or it is having like a protein structure, or it may be amphiphilic in nature. So in which cases what will happen, you can surface conjugated drug also we can make it. So there are the 3 sites which are making it, and most of the times we will make the gene therapy by entrapping our moiety, because the protein is which is having a lipid soluble in nature. So it is having a lipophilic, that is a hydrophilic in nature. So they will be entrapped within the hydrophilic region of your liposome, whether it is a DNA, RNA, whether it is a small interfering RNA. So to interfere or changes your function of gene. So you are having a mRNA, so you are inserting there and giving it. This is one site. Another one which you are going to have is not only this DNA and RNA, we are going to have a protein also. We can just surface ligand the particular moiety, whether it is an antibody or it may be a peptide, it is a small molecule like a drug. So we can endcap it. At the endcap it, we will be making it an activation. With that activation, it calls the covalently bonded or hydrophobic and enough density bond angle and length, so that it will be having a sterically without any problem reach to the target site. So such moieties which we are going to give on the surface, and ultimately at the end, even all these approaches are not successful. We have done all of them attempted. One is within the cell, the drug which is having a small molecular weight on the surface and on the within the layer itself, or it may be in the form of DNA and RNA. So we are making it, so we are not able to successful. Then we are using stimulus induced. So in that we have already got the four types of approaches in the stimulus induced, and we are at the surface. We create this stimulus to get a response. So one is a particular ligand that will be having a response to the pH changes. Another one is a specific enzyme, changes in the specific enzyme which is having a specificity. Or another one is it is having a redox principles and changes which is having a specificity. Another one is the light. So these are all the things which will be making it to act specifically in a particular site. This is ultimately we need to use it if all the responses are failed in our liposomal designs. This is a little bit we need to adopt a sophisticated technology so that we are having a success better. So these wide designs which are making us to give wide variety of the drugs to reach the target site. This is actually what our journey. So you need to write a diagram if you want to get good marks in your exam. So let us try to memorize that. So you need to write it. And in this journey, we have got so many problems, so that we need to add not only the liposomal excipients which are required for their formation, we need to use a stabilizing agents for these gene products, because we need to stabilize the DNA or RNA or any aptamer or any nucleic acid sequence or protein, that is a amino acid sequence, or it is a basic drug, intact. And those chemicals should not be toxic. So for that purpose, there are so many list of the classes have been given in the literature. This slide is giving you idea about it. So I kept in this. It is a big list. It is present. You just read them after the class is over, so that you will be able to understand what are the other excipient adjuvants I need to add in the targeted drug delivery system, so that I can solve the problems of the delivery. This is what you need to understand. See, if in this particular list you have got amino acids, antioxidants, chelating agents, fatty acids, proteins, metal ions, and polyhydric alcohols, polymers, and surfactants. These class of excipients, these adjuvants are being used, and different chemicals are already present in the grass list. It is presented there to serve the different purposes, like either it is a solubilizer, or it is acting as a buffer, or it inhibits the isomerism. So like ascorbic acid, you want to make some enantiomer which is having a Lexco or Devo in the case of basic drug moieties. So they are stereospecific. Then we need to use this aspartic acid as the inhibition of the isomerism. What concentration we need to use? We need to search in the literature, and we need to find how it is going to affect the stability of it. We need to fix that concentration. Another one, you have got a thermostabilizer like a glutamic acid if you are having any changes in the degradation with respect to the temperature. So we are going to have thermostabilizers also. And we are going to have aggregation prevention, because when you are having aggregation, then what will happen? You are going to have a bigger sized moiety that will have a different behavior. So you should not make that aggregation and forming like a clump in the granule, what you are observing it. Such things we should not be present here in the nano level, because it will take to the journey from nano to micro level when it is having an aggregate. So we need to add a leucine like amino acid to prevent that. So you have got antioxidant to prevent the oxidative degradation and the protein conformation changes you not to avoid. EDTA salt to prevent the degradation by the oxidative changes by in the presence of heavy metals. So they have the, most of the times you will be having a free radical scavengers. These EDTA salts serve that particular purpose. So that will prevent the chain reaction to keep the end. So this one, it is required to be done by the EDTA. If you see further, you have got a fatty acids, and they serve as a stabilizer, further enhancing its stability and shelf life. And you have got a proteins, which will be having a surface absorption of a non-required moiety junk onto the surfaces. So they will be having a specificity and which is having a complexing agent and the cytoprotectant. Cryoprotectant means if you are using a freeze-raised product, so this liposome should not be getting damaged under those conditions. So sometimes you require to give as a reconstituted product, you need to have a that is the lyophilizer, that is a called as a freeze drain. In those cases, you need to use a cryoprotectant. So when you are freezing also, the liposomal structure should not be disturbed. So such cases you are using a serum albumin. You are finding in liposomal preparation most of the times all these components, they are little bit costly when compared to your regular chemicals. That is the reason even a small bottle which is having cost of 5 to 10,000 rupees. So it is required to be done in a very cautious manner in our lab if you want to do these targeting approaches. And also we require to find the surface and the particle sizes, we need to use electron microscopy. That is the reason these are all little bit costly approaches and not possible by the regular labs, even though they are appearing as a promising. And unless otherwise we have got a strong determination and little bit encouragement, then it is a little bit tough to achieve that, because it is in the midway. We can't find whether we are reaching in a correct site or not, whether we are having a correct path or not. So the metallic ions like a calcium and nickel or magnesium, manganese, they stabilize the protein conformation because we have got a tertiary structure. The structures like a primary and secondary tertiary structure should be preserved to help the protein function. Not only that, we have got a stabilizers like a polyhydric alcohols. We need to understand their function as a multifaceted, because we have got a multifactorial design. We have already seen previous. Why is this multifactorial? If you see all these components, the list is getting piled up more and more, and we are having a lot of complexity to select the what in the list, what class and what agent and how much concentration. For that purpose, we are employing a design of experiments, and we are seeing them. And in this case, you have got so many varieties. So many varieties like ethylene glycol, it is acting as a stabilizer, and glucose, it strengthens the conformation, and lactose, it is also a stabilizer, and mannitol, it is a cryoprotectant, and it will give the bulkiness, and it is also having a capacity to create the cake formation, because for the reconstitution you want to have a cake. So if you want to prevent the degradation of the chemical which you are using as a drug is hydrolytic in nature, so you cannot dispense your injection in form of pre-filled, or it is in the form of liquid dosage form. You need to give a freeze-dried product which is already containing a cake. If a material is containing very less weight and it is not visible to your naked eye, in that case you need to have a diluent. So in that case you need to have this mannitol. Another one is it is also having a cryoprotectant. Propylene glycol, it is having a prevention of aggregation. Sorbitol, it also prevents the denaturation and aggregation. Sucrose serves as a stabilizer. So the trihalose, most of the times these polyhydric alcohols, they will also be incorporated in addition to the polymers. Like PEG, we are having a reticular endothelial system escape. We are going to use it, and povidone to prevent the aggregation. Surfactants, obviously we need to use Polaximir 407. It has been already studied in the literature. You find so many articles. They prevent the denaturization and stabilizes the cloudiness. So they make it very clear and making it in the form of a scaffold, and make it dosage form very long acting. That is stabilizes more. Polysorbate 20 and 80, they retard the aggregation. So we have got a different wide variety of agents. We need to pick up one, and we need to fix the concentration, and finally we need to adapt in the final optimizer formulation. So this is actually what we are going to have as a liposome specific to get a success in the drug targeting and nucleic acid delivery. Now we will be entering little bit deeper now. We have already seen one dosage form with respect to all these things. And these are all tiny in nature. I don't think so you will be able to see now in the mobile screen. What you do, I will be posting in the Google Classroom a little bit later, and You download it and a little bit zoom it so that you will be able to find this list. Because the biggest list, if I take it individual and in a zoomed version, so it takes 10 slides like that. So I thought, okay, let it be you yourself downloading it. It is available to you in a little bit later. Then what are the potential target diseases for the gene therapy? This is another question you are going to get in the exam. What are the, I am making a journey. I don't understand what is the gene therapy, a little bit further understanding later also. So what are the potential target diseases? What diseases I can treat? So this is actually what is important so that I can make my journey started by the extrinsic approach or intrinsic approach. So I will take a disease and I will find the treatment. For that, can I use this concept of a targeting drug delivery and nucleic acid drug delivery? There is another one is I will take a drug and I will make that particular drug to available to the system by making a dosage form. We can any drug which you are taking it and going from the bottom to the top to reach there. So what you are going to, you want to attack the disease by taking a disease and finding different drugs with it. And I'm taking a drug, I'm finding different categories of utilities of that particular drug and delivering it. So in either case, what are the target diseases we are going to have? In either case, we need to ultimately treat the disease. So for that purpose, if you look into the gene therapy, we need to don't misuse our technology. Generally, if you misuse our technology, what will happen? The patient will pay very higher amounts of money for the lesser cost, and the cost benefit analysis is not much there, and value of the system is not correct. So it is not having much value. So we don't want to misuse it. Then what we do? A particular journey in the gene therapy. So what is the particular site of action for the gene therapy is the chromosome. So it is a basic unit of the heredity carried out on the chromosome. So that we are going to attack now and treat the disease applicable to this particular chromosome. Then we are calling it as a gene therapy because the gene is nothing but a chromosome. So the pairs of chromosomes, which is holding entire our genome and making the functionality of the body and making the uniqueness and identity. So that is actually what we are going to have a target, potential target. It means our single target, if you can say, is simply for the gene therapy is your chromosome. So in that particular case, even entire chromosome I need to attack. So what is my potential target? The specific sequences of the bases only I need to attack. Don't get confused that the entire chromosome I need to attack means you are altering your species and changing it and making him die. So it is not our requirement. We don't want to make a human being into an animal. It is something, it is not required also. That is the reason so many world regulatory agencies, they are controlling the research organizations not to do such methods and trials. So in the view of getting a specific capabilities and the skills, and don't do that because it may cause a lot of damage to the nature. So because we are handling a basic unit of heredity, so we don't want to make entire genome is getting changed, chromosome, but it is a specific sequences of the bases. So you know that there are so much of research which is being made, and there this entire genome is getting altered to have a here got that is attached onto the top of the rat. So if you are seeing these studies, it is appearing like peculiar in nature. Why do you want to insert the ear of a human being nature into the rat surface, that is the skin? So is it actually, it is only curiosity. If I'm inserting the particular genome, which is having a capacity to generate the ear, if I'm putting the genome into the rat, what will happen? Obviously, it will be getting ear onto the rat surface, skin surface. So it means that the utility is, it is appearing that it is functioning, but what is its use? So we don't want to make the all the rats which are born subsequently, it is good carrying a ear-like structure, ear lobe onto the exterior of the rat. But it is not actually the correct research. If you are doing it, we need to attack a specific sequence of the bases that encode instruction how to make a protein. So we have got a problems in the proteins, whether it is any growth hormone or it may be any erythropoietin, or it may be insulin, or anything. Such proteins are only which is causing a lower or a higher purposes. So we have got to attack them. So that sequences only we need to see. This is actually what the target of our particular thing which we need to see in the gene therapy. Another one is mutation of the gene results in the failure to synthesize a particular protein because it got already mutated. The mutation, it may be either it is a permanent or temporary damage which has occurred by the external stimuli that causes the changes in the alteration and changes in the functioning of the particular genome, that is the chromosome. So in that particular mutation, the small segment of that particular chromosome is getting altered because of the external stimuli. Maybe it may be a radiation, or it may be a chemical, or it may be anything which is relevant to the mental, that is belief or biological. So that causes the mutation, that failure the synthesis of a particular protein, that we need to attack. Another one is what we need to attack. The results in the most of the times the genetic mutations, they result in the abnormalities regarded as a genetic disorders. Those genetic disorders only we need to consider for the gene therapy, like hemophilia, night blindness, the heredity which is appearing as a common factor for the maybe diabetes, or maybe a neurological problems. It may be Alzheimer's, it may be Parkinson disease. Such as genetic related disorders, which is nothing to do with the external stimuli by the offsprings of the next generation bound to get, because the previous parent one they are having already such disorders. Such things we are going to have as a potential target, so that we are using a correct usage of technology for the humankind benefit. So a gene can be delivered to a cell using a carrier which is called as a vector. So this is what we need to understand. So this vector we have already seen previously a little bit before 1 hour is a liposome. That is one carrier we have already seen. Don't get confused with that. Don't see that I need to fix for only liposome for my gene therapy. So there are different things are available. First we need to understand the concept of gene therapy, then we can choose it, whether it is liposome or anything else. So what is the concept of gene therapy? An experimental treatment that involves introducing the genetic material like your nucleic acid into the person's cell to fight or prevent the disease. There are two things. We know that the disease is going to cause it to the next offspring because it is a genetic disorder coming from the heredity. We prevent it in advance. It has already occurred. The person has given birth. We want to alter it. Then we need to have a fighting the particular defect. This is actually what we are going to do in the gene therapy. Another one, it is used to correct the deficient phenotype so that the sufficient amounts of the normal gene products are synthesized to improve the genetic disorder. It is not that which is lacking it, but it is having a deficiency in those functioning, so that we are correcting it. This is one we are going to do it both for the prevention and the cure. So this is actually the basic concept. It means now we do what type of steps we need to do in the gene therapy. So this is very important and basic concepts of the gene therapy. So the steps involved in the gene therapy include identification of the defective gene, that is the first step. In the chromosomal sequence, what is the one which has got a defect, so that we need to identify it, because we know already human genome project, each genome segment and the particular part has got specific function. The defected portion in that particular genome, we see it and from the gene database, and we are able to find out the defective gene. And the cloning of the normal healthy gene, we take it and we clone with the segment which is having a normal, and we are going to identify the target cell or tissues where we need to have a alteration, and insertion of this normal functional gene into the host DNA. This is actually what the journey is. So we are taking a identification of the defect and finding the normal one, and we are making it made outside, and identifying the target cell where the defect is present, and insert this normal function gene into the host. These are all the 4 steps, major steps which are present in the gene therapy. So now the question is identification of the defective gene. We can know according to the pharmacodynamic functions, that is the biological functions of that particular body. we can link those functions to the specific segment, and the defective gene portion we are able to see it, because of this portion is getting defected, and because of the defect we know that what are the alterations which are already been present in the genome database in the functions of the diseases. So we will be able to see that yes, this is actually the defect portion. Then what we do, we take a normal one, and we will take that particular genome, and we insert the normal portion, insert in that particular defect portion, and keep it ready. And find out whether the target cell, whether it is a stem cell, or it may be a bone marrow, or it may be a particular tissue. So where we want to make this particular cloned gene available. Then we will use the delivery systems to insert this normal functional gene into the host DNA. So it means we have got identification, which we have got a simulated techniques, we have got a cloning of that one, we have got a laboratory techniques, and we know the identification of the cell which is having a particular specific part where it is having a defect, we know, and we can easily say that where the particular portion we need to target. So it is already known to us, and we are making this available. For this purpose only, we are going to use a delivery. If we are having a formulation scientist, at this particular stage, we are entering it. That is insertion of a normal functional gene into the host DNA. It is our job in the steps involved in the gene therapy. Most of the times, we are having a different vectors for using this particular fourth step, which we want to get completed. So how it works if we are having these all the steps which are going to have it? What will happen is a vector delivers the therapeutic gene into the patient target cell. A vector. So we are using a inert, and it is not having any harmful to the body by itself. Even if you are giving a blank to that, it will cause any damage to the host. That is actually ideal vector is. So the target cell become infected with the viral vector. Once we are giving that particular vector, whether it is a viral or non-viral, it is getting affected or getting diffused or getting entered with this particular vector. The vector genetic material is inserted into the target cell, and functional proteins are created from the therapeutic gene, causing a cell return to the normal state. This is actually what the journey is. It is present. For that purpose, we are using a vectors. So what are the different types of gene therapy which we generally adopt? What are the different available techniques? So this part we will be seeing in the next class, and a little bit advanced we will be going a little bit later. So whether it is for the cure, or it is a treatment, or it is for this generation or for the next generations, based on this only we are going to take it. Based on that particular principle, we select a vector, whether it is a viral or non-viral. So a biological vector or non-biological vector. So we are going to decide that. Then we are going to have excipient selection. We have got a adjuvant selection. Then make the suitable delivery system and trials in a primitive animals. Then giving to the advanced levels where the treatment is there in the next stage. Most of the times, the journey which will be given as special permissions from the regulatory authorities, because they will be generally they are all called as orphan drugs. They are present very less population in society. Maybe 1 in 1 lakh, like that they will be having these patients like this. It is not a regular one. Even they don't consider like that. It is orphan diseases only we need to employ. That in nearby future we may be employing each and every disease, having a reason to have a defect at the molecular and cellular level. So we want to treat them at the cellular level. So we need to use these steps. There is no other option for us. Even a simple painkiller we may be giving a little bit later, maybe this approach only, because we don't want to make entire body suffered with the 60 kg body weight of our human being. Should not be suffered for the 10 gram body weight of a tissue which is present as a defect in our body. So this is actually what the journey it means. We will be employing sophisticated these vectors. Maybe a nearby future for the normal delivery also we are having a little bit of specificity there, so that we will be having a better efficacy and the advantages than over the conventional dosage form. So we will see the types of gene therapy and vectors present in the gene therapy, and we will see other gene therapy for the cancer, and we are going to use how the approaches are being used. So this journey will be keep on going. So we will meet in the next